Okay, so let me start by saying these are some tough acts to follow. I have, as you can see here, some videos, but they have no music. They also don't feature any of these explosion views of body parts. So in comparison, they are pretty lame. And then also, Johanna just pretty much preempted all of my introduction. And then also, even though she was too modest to mention it, but all the beautiful data that she just showed was published in Cell just a few months ago, which I think is the highest profile paper that has come out of FaceBase 2 so far. So really amazing work. So I'll try my best. Let's see. So before I start, I can actually see this. I'll use this thing. I brought Eros and Kaylin from my lab. They're somewhere back there. They're both postdocs working on our FaceBase 2 project. Eros more on the computational side. Kaylin does most of the ChIP-seq work. And they have posters on which they will expand this afternoon on some of the aspects of this study or of our work. So I'll start with a little bit of background, setting the stage, and remind you of what we're trying to do and why. I'll then give a brief progress update regarding data generation, in particular data generation from mouse facial regions and from actual human craniofacial tissue. Talk a bit about enhancer validation and characterization and OPT. And then I'll talk a little bit about ongoing analysis efforts, interactions with other projects and collaborations. In particular, I'll talk a little bit about integrative analysis of human and mouse chromatin data that we've generated so far. I'll show 2 slides about our interactions with Johanna's lab, and then I'll show you just one example of how we are trying to act as a resource for investigators beyond FaceBase 2 for the investigation of enhancers. So a little bit of background. So again, our lab has for a long time been working on distant acting enhancers, similar to what Johanna described. And this cartoon is just here to remind you, very generally speaking, what these enhancers are. So when you look in this works for all sorts of developmental processes. So here in this cartoon, you see a gene that is expressed in the brain and in the limbs. And then what we frequently see in vertebrate genomes is that these genes are regulated by these distant acting sequences that can sit far away from the gene and act on the promoter to drive different parts of this expression pattern. So what happens at the molecular level is that these enhancers are short sequences that have recognition sites for transcription factors. These transcription factors bind, coactivators bind, then a few other things happen that are not shown here. Importantly, certain modifications to the histones happen here, and then this whole complex loops around to the promoter and activates transcription. And in different cell types, the same thing happens, just with a different enhancer and a different set of transcription factors. So in the brain, you would find this kind of conformation. And what is shown here for completely different body parts is also true for craniofacial development. We know that many genes that play a role in craniofacial development are regulated by different enhancers that drive different parts of this. And this is really the goal of our study, to provide better resources for mapping and understanding these enhancers, because at this point, it is well understood that beyond the genes themselves, variation in these enhancers plays important roles in evolution, as we just saw, but also in disease processes and in general developmental biology. So to look at craniofacial enhancers, what we like to do is, rather than cell-based models, we like to look at primary tissue. So what we do is we take actual craniofacial tissue from a developing embryo. Shown here, this would be a mouse embryo in this example. We take the entire face region, and then we use ChIP-seq to map various chromatin features. In this case, this is an antibody against P300, but we can also use antibodies against all sorts of histone marks. And this generates then for this particular tissue a genome-wide map of all these little peaks when you map these reads back to the genome. And all of these little peaks that you see across the genome are then candidate regions for enhancer activity in that particular tissue that you're looking at. This generates genome-wide maps, but we can also take any of these peaks and characterize them in more detail. We have a transgenic mouse assay for this purpose, where we can couple any such sequence taken from the mouse or the human genome to a LAC-Z reporter. We generate transgenic mouse embryos with that, and then we look at these embryos to see where exactly the enhancer is active. And this is really important for two purposes. One is these chromatin-based methods are not perfect. You get false positives. So you want to validate that it's really a reproducible enhancer. And then on top of that, the spatial resolution of these is much higher than what you can dissect. And so just to show you a few examples, these are some enhancers that we identified during FaceBase 1, where we had a project that used similar methods. So you see here different enhancers, and even at this low level of resolution, you can probably appreciate that the activity is in different parts of the developing phase. But each of these patterns is highly reproducible. So if we repeat the experiment, build another transgenic, we see the same pattern over and over again. And so this tells you really how reproducible these enhancers are. We can also, to get even higher resolution data, OPT data, optical projection tomography. We do this with David Fitzpatrick's group. And you see again here, these are just 6 still images. Behind each of these is a proper 3D model, and you can access this data through FaceBase. So what happens here is these like Z-stain embryos get scanned, and then it gets converted into a false colored image where all unstained parts of the embryo are shown in green, and then the enhanced activity is shown in red. And then by looking at these, you can get a really high resolution view of where exactly the enhancer is active. So in FaceBase 2, what we're trying to do is 2 things. The first important one is generate higher resolution chromatin data from the developing phase. And we do this in 2 different systems. We do this in mouse and in human. In the mouse, we are trying to dissect out different parts of the developing phase at critical stages of phase development. So E11.5, 13.5, and 15.5. So these different colors indicate the level of resolution that we're doing this. And we are also looking at actual human fetal tissues. These are from Carnegie stage 18 and 22, which corresponds to 44 and 54 days post-conception. Here we are more limited in terms of the availability of the material, so we get the entire phase region, but it has the advantage of being able to perform RNA-seq and ChIP-seq on actual human tissue, which gives you a lot of additional information, as I will show you later on. So in terms of the assays that we do ChIP-seq. We don't do P300 this round because it turns out to be technically challenging. Instead, we look at a panel of different histone modifications that mark promoters, enhancers in their active and poised state, and repressed chromatin. And I'll show you the actual marks later on. We also do RNA-seq to get matched transcription profiles. Again, the level of spatial resolution here is lower than what some of the other projects in FaceBase 1 and FaceBase 2 have been doing. But the main purpose of this is to get RNA-seq data that comes from the exact same preparation from which we have the chromatin data, which is very important if we want to match these different types of data. And then as another addition, we have a TAC-seq. So this is not part of our original proposal. It's technically not a milestone that we have to do, but it requires small amounts of input material that we have anyway, and it provides very nice data. So we decided to do that also for all the samples that we get moving forward. So here's our data generation progress. So during year 1, I show, and this top table, I think I showed you most of this last year already. We looked at E11.5 and generated RNA-seq, and then K4ME1, which is a histone mark that is found on enhancers both when they're active and when they're just in a poised state, so not quite active yet. K27 acetyl, which is probably the most important mark for enhancer prediction. It's supposedly associated with enhancers only when they are active. And K27 ME3, which is a mark for repressed chromatin. And we have now in year 2 focused on E13.5. So we have now completed data collection for all of these different marks from these different parts of E13.5 mouse phase. And then we have also processed many additional human tissue samples. So each of these tissue samples is derived from a single human fetus where the whole phase region has been dissected out. And you see here the stages. So our two main stages are CS18 and CS22. Occasionally, we get material from stages that are slightly later or slightly early. We see here 19 and 23. We also process those to get some data for these intermediate stages. And what you can see here is that we have now a nice K27 acetyl data set, probably the most important mark, for about a dozen or so samples. And then we're starting to fill in these other data points. We have RNA-seq for most of them. These ones that say S means we're still sequencing the data, but hope to have that soon. Then here, these are in progress, ME3. We have a TAC-seq for quite a few of the samples. And then the one that has given us a bit of problems initially was this one, K4ME1. So for these, where you see the T, we're still troubleshooting these, but we're making progress towards getting the data for those too. Something that Kaylin has spent a lot of time on during the last year is improving our ChIP-seq protocols in terms of the data quality. This is particularly important for the human samples because you are limited in terms of how much sample you can get. What you see here on the left is data from year one. And I showed you some of this last year, so we didn't do a TAC-seq back then. Here you see K27 acetyl. You see some peaks here, like for ME1, a little kind of suggestive peaks, but not great. And then this one, K27 ME3, with again some peaks, but a whole lot of background. So with improved protocols, we get now cleaner data out of this. You see here data derived from a later CS18 sample that was done more recently. See a few things. So first of all, so this is the same genome region and the two different types of data sets. First of all, of course, these big peaks that stand out here, even with the lower quality data, stand out nicely here. But what we start to see is that this is only suggestive here. They also get K4ME1 peaks here, and then this is also mirrored up here in the ATAC-seq data. At this level of zoom, it's not quite evident, but generally the ATAC-seq also has slightly better spatial resolution along the chromosome, so it can help to pinpoint where exactly the center of the peak is. These ChIP-seq peaks, even though they look sharp here, when you zoom in, they tend to be a bit wider. And then in addition to confirming these, we start to see new peaks. You see, for example, these two here, which you don't really see in this lower quality data. And then importantly here, for the K27 ME3, we now start to see nice peaks that clearly stand out over the background. So moving forward, we are using these improved protocols, which will hopefully increase the value of the data for the users of FaceBase. The other important component of what we're doing are these transgenic assays. I explained briefly how it works. So we can take these peaks and generate transgenic reported data, and we use this really for two purposes. One is we can use this to look at specific genomic loci that we are interested in. And these are often collaborations. A typical case is someone from within FaceBase or outside collaborators come to us and say, well, we're working on this locus. We have either developmental biology data or we have human genetic data pointing to this region. Help us find the enhancers. And then we work with these groups to go through our ChIP-seq data sets, identify the most likely regions, and then can take some of these and put them in the transgenics to check which of them are really active, and if so, where exactly are they active. And I'll show you an example of that later on. The other purpose of doing the transgenics is that I mentioned that many of these chromatin-based prediction methods are not perfect. So when we generate these new data sets, we want to get a sense of how good these predictions really are. So we are also using this to test general predictions for new chromatin data sets to get a sense of the success rate. Over the last year, we have performed 26 transgenics experiments. I don't have the time to show you all of them, but just want to briefly mention that in about 2/3 of the cases, these were positive for some kind of craniofacial structure. As in FaceBase 1, I want to point out again, so part of these transgenic capabilities are specifically set aside as a community resource. So if you're one of these people who are working on a specific gene of interest and you want to find enhancers, please come to us and we'll be happy to take a look and help you with transgenic work. We've also generated more OPT data. So last year we didn't have a whole lot of it because this always lags behind a bit because we do this together with David Fitzpatrick's lab. We start to get the new data sets in, many of these generated during the first year of FaceBase 2. You see some examples here. When you go to the FaceBase hub, you can currently find 43 OPT data sets. These are mostly older data sets. We're currently working with the hub to get these new data sets integrated, and we may discuss this later on in this meeting. Then hopefully also the OPT viewer will hopefully be able to accommodate these, including the red and green color images soon. So a little bit of analysis, interactions, and collaborations. So what can we actually do with the data? So one important rationale behind our current FaceBase project was to get these sub-regional patterns, in particular from the mouse face, with the goal to do something that is kind of similar to what Yang showed earlier on. So you want to get data from the different sub-regions and then be able to look for differential peaks. So essentially the equivalent of doing RNA-seq for differential expression, you would look for differential peaks. And when you look at the raw data, you see that there are several thousand of these peaks, in particular from K27 acetyl, that show a significant enrichment in one region or the other. And in principle, these should give you a way to predict where exactly the enhancers are active. I showed some early data about this last year, and the bottom line was it works to some extent, but it's not perfect. When you test these, oftentimes the pattern that you see in the transgenic doesn't perfectly match what you would have expected based on the K27 acetyl alone. So what we're currently doing is trying to integrate these data with other types of data that we have available with a goal to be able to make better predictions of where exactly an enhancer is active. And we do this in a number of ways. One important way is to intersect these with the RNA-seq data sets that we generate. So data generated from the exact same tissue preparation to get a sense of where the presumptive target gene is expressed. The other important thing that we can look at is a transcription factor binding site. So we can perform computational motif analysis of these enhancers, and then we know where most of the relevant transcription factors are expressed within the developing phase. We can tell from our RNA-seq data, we can go into sequence databases such as, expression databases such as JAX, and then intersect all of these data sets to improve our prediction. And this is work in progress, but let me just show you the general idea here. So what we do is, and this is again highlighting just one example, but we're trying to do this systematically across the entire genome. So what you see here on the left is one genomic locus. It's probably hard to read, but this is PATX1. And what you see here at the top is the chromatin data, so ChIP-seq data for K27 acetyl, which sits on genes when they are expressed, as well as on enhancers when they are active. And then down here, you see the RNA-seq data. And so in each case, I'm showing you 4 different components: medial and lateral nasal process, maxilla, and mandibular. Here, the same 4 regions. And then for the RNA, you also see the plus and the minus strand of the DNA. And what you see here, as expected, is that PATX1, the actual exons, are transcribed, in this case from the bottom strand, because PATX1 is facing to the left. And you can also, as expected, see here the ChIP-seq signal. And both the RNA-seq and the ChIP-seq signal is pretty much restricted to mandibular process. And now, this signal here is just sitting on the gene, so that's expected, but we want to find the enhancer. So what we can do then is we can look for other ChIP-seq peaks that are nearby that show a similar kind of tissue specificity. And indeed, when we look here quite a bit away, so about 50 kb upstream in the intron of a neighboring genes, we see these different peaks that show the same kind of tissue specificity. So this would be a great candidate for enhancer activity. And then here's a similar example, slightly more complex. This is near the ALX1 gene, which of course is also involved in craniofacial development. Similar situation, except that the pattern is slightly more complex. You see here, when you look at the chromatin data, that both in the medial and lateral nasal process, there's a ton of signal sitting on the gene, just a little bit of signal and even less signal in maxilla and mandibular. You see this reflected here in the RNA data. You can also see that in the nasal process, there's a little bit of antisense transcription coming from here, but the really interesting part is up here, marked by these red arrows. These are regions that are, again, a few kb upstream of the actual gene. They show the same kind of tissue-specific activity as the gene expression itself. And so these are, again, candidate enhancers. And so this is something that is currently in progress. We're trying to do this more systematically, and we're moving these candidate regions forward into the transgenic. So including these three shown here by these red arrows, these are currently being tested in transgenic to see how well this really works in reality. A few words about human-specific craniofacial enhancer. And Johanna provided a little bit of background about this. So just to give you a sense of what we can look at from our end. So we have human and matched mouse data from actual tissues, and this has advantages and disadvantages. The disadvantage is that it's a slightly messier preparation because in reality there are, of course, multiple cell types in there. At the same time, I think it's clear that there are certain complexities of these developmental processes that are insufficiently modeled in these cell culture models that you can do from human and chimp. And so what we're looking at is tissue data. And what you see here, so to guide you through this kind of complex matrix, you see here on the left ChIP-seq data for K27 acetyl, which is the enhancer mark, derived from 3 different human specimens. And in each case, so each of these thin lines is the same region of the genome shown 3 times. And then you see in total about 2,000 thin lines. Each of these is one different presumptive enhancer. And what you see in each case is here that the same enhancer shows a reproducible peak in all 3 human specimens. So it's reproducible. What we can then do is we can look in the mouse genome and ask the question, which of these are conserved? So we can map all of these human intervals over to the mouse genome and then look at our spatially resolved mouse regions, here again the 4 regions, and ask which of them show up. And what we would expect in a perfect world is if they're conserved, that they would show up as a peak in at least one of these 4 subregions. And it turns out that in about half of these cases from the human, when we lift them over, we do see significant peaks in at least one of the mouse tissues. So that means these are presumably functionally conserved, or at least the chromatin signature is conserved. But then we also see a fairly large number, about 1,000 or so in this initial data set, that showed no signal at all in the corresponding mouse region. So these are human-specific peaks. And when I say human-specific here, I mean they are present in human, but not in the mouse. We can't do this for chimp, but these kinds of data sets can be very helpful if we start to look, for example, at Johanna's human and chimp data to make a guess which of the 2 is the ancestral state. And then there's some number of human peaks that cannot even be lifted over to mouse. So these are sequences that simply don't exist in the mouse. And as Johanna mentioned, we've worked quite a bit with them over the last year or so. So we had several joint group meetings and phone calls to try and integrate these different kinds of data sets with the goal to find additional examples of enhancers that have presumably contributed to human or chimp evolution. So from Johanna's group, there are these human and chimp chromatin marks, also RNA-seq data and some ATAC-seq data. And then we're trying to integrate these with our tissue-derived data sets. And then at our end, we've also done some additional analyses for these data sets where we're trying to bring in MGI known phenotypes from mouse and some analysis related to topologically associated domains, which can help you to figure out what the target gene of a given enhancer is. And so from these efforts, we've come up with a combined list of about 20 sequences that are currently moving forward in transgenics. This is in progress, so I don't have a lot of data yet, but just to give you an early flavor of this, this is one of the sequences that is in progress. So this is a sequence that was predicted based on the chromatin data to be a craniofacial enhancer in human, but not in the chimp version. So we took the human version and put it in the mouse. And you see here mouse embryos carrying the human enhancer. And we don't have OPT data yet, but hopefully even from these images, you can see that they have reproducible expression here in the midface region. And the chimp experiment is still in progress. In a perfect world, okay, I have to move on. Perfect world, it will not have signal. And then I won't go into detail for this one then, but we're also working with outside investigators. This is just one example. I'll talk through the details. This is with Seb Dworkin at Monash University in Australia. This is just one example of an investigator who approached us with a detailed developmental biology study, and then we helped him to look for candidate enhancers that might drive this gene, and we found this particular one that has a pattern that makes a lot of sense, looks really interesting. We have now OPT data for this that confirms that this is expressed in the ectoderm and particularly strongly in the facial prominences as well as the limbs, and we're currently following up on this in detail. And I think I already went over time, so I won't read out all of the collaborators, and I'll just leave this up here while I take questions.
Who's got a question? Oh, please, yeah. Anybody have any questions for Axel?
Could you show me again that slide when you compare the human enhancers against the mouse enhancers? Please. So yeah, in the bottom, when you compare the orthologous mouse regions, for an example, if you get the enhancer sequence and you blast it against a matrix database, you think that it's possible to find a correlation in between like conserved proteins that are not there in mouse or suffered some kind of modification? And this is why when you look for DNA sequences, you cannot compare those.
So you're referring to these or to these?
Comparing the left side and the right side.
Yes.
So you see on the right side the orthologous mouse regions, but you cannot find something in your data, right?
Yes.
But in the upper column, you can when you compare both, right?
Yes.
So if you take just one sequence from one enhancer, just one, like 1,000 bases, and you use you blast it against matrix databases to find those transcription factors that could bind to these sequences.
Yes.
So,
well, you could probably find another enhancer that has a similar binding site content, but this is not this experiment. So what you do here is this is based on lifting over to the mouse genome. So this is not a simple blast search. This is identifying the truly orthologous region. This is based on whole genome alignments, right?
I got it.
And so the question that you're asking here is the actual sequence the sequence conserved in that position, and is the chromatin pattern conserved? You might, there's clearly evidence of things such as enhancer turnover, binding site turnover, and so on. So in those cases where the enhancer is not present down there, you might have another enhancer somewhere else in the genome that compensates for that, if that's the question. However, I think...
Yeah, thank you.
Okay.
Hi, Axel. So I kind of have a similar question, I guess, and I'm just surprised. It looks like, if I understood what you just explained correctly, that something like 18 to 19 out of 20 of your attempts to lift over, that is, find the truly orthologous position and sequence, succeed. I find that very surprising. When I've tried to do it, certainly our success rate is much lower, and the difference between mouse and human in terms of gene duplications and things would suggest that 1 in 20 might be low.
When you try to do that, are you looking at random sequences or are you looking at regulatory sequences?
No, we're looking at regulatory sequences.
Okay, so something that is well understood is when you just perform ChIP-seq for whatever P300 or K27 acetyl and find these enhancers, they tend to be highly conserved. This is something that has been observed many times, and so compared to genome-wide average, you would expect to find a much larger proportion of sequences that can be lifted over. Now, the exact proportion that lifts over will depend on how conserved that data set is that you start with. And I suspect when you say regulatory sequences, you are referring to some other way of assessing regulatory sequence.
But I would also submit that what your success rate is also defined by the parameters by which you define success. And so your criteria for homology, for example, determine outcome. So how sure, what sort of external validation do you have that you truly are finding the homologous element?
So with this kind of analysis, so for all the liftover, this is using the standard liftover tool that has been well established for finding orthologous regions, right? And this is not a blast. This aligns usually megabase scale regions between mouse and human. So I'm looking at this analysis. I'm less concerned about the accuracy of the liftover. I think there are other experimental factors here that would be much more concerning, that are a bigger limitation of this kind of analysis. For example, that this kind of analysis assumes that you're looking at the exact same time point in both species, which is almost impossible to do because it doesn't develop completely collinear and you can't really catch the exact time point. Also, all kinds of analysis of this type are limited by the quality of the sequencing data. So, for example, this down here, these guys might include some cases where there is a little bit of signal, but it was just not significant enough to be called as a peak.
Yeah, so one rather technical question, and then if there is time, another question. But so when you test in transgenesis these enhancers, do you take the smallest fragment that contains the enhancer signature, or do you extend a larger fragment to test it in transgenesis? Because it appears that the readout can vary quite substantially according to the two different approaches.
It's the latter. So typically, we don't restrict ourselves to technically what is called as the peak, but we include some flanking sequence. Most of these constructs that we test are between 2 and 5 kb in size. Yeah, that said, in many cases, you could cut them smaller, but our main goal in the initial attempt is to see the enhancer activity. And so we rather make it larger than smaller, and then refine as necessary with additional follow-up experiments.
From the last paper also of Francois Spitz in DevCell, it seems that if you enlarge the fragment quite substantially, like in the order of several kilobases, you lose some detection of some activity versus taking the isolated enhancer, the shortest fragment. But anyway, do I have time for another question? Comparing the ChIP-seq data to the RNA-seq data that you intersect, I'm curious to know how far or how close is the gene for which you find differential expression by RNA-seq near to the enhancer that you find by ChIP-seq methods. So how far do you extend your, saying this is the target for this enhancer?
I can't give you a single number what the average is, but something that we've been doing a lot recently is using TAD data, so topologically associated domains. Mm-hmm. These seem to be a good predictor of the boundaries of a domain within which an enhancer can act on a gene. So this is what we've been using a lot recently. And we're still trying different approaches, but right now this seems to be the most promising one.
Okay.
And these can be up to a megabase in size, as you know.
Okay, great. Thank you, Axel. All right. Okay.